So this last section talks about taxonomy. Taxonomy is the branch of biology that names and groups organisms according to their characteristics and evolutionary history. Organisms were first classified more than 2,000 years ago by the Greek philosopher Aristotle. Organisms were grouped into land dwellers, water dwellers, and air dwellers. Plants were placed into 3 categories based on the differences in their stems. As new organisms were discovered, his system became inadequate. So Linnaeus, a Swedish biologist, established a simple naming system for classifying and naming organisms. He developed a hierarchy for organisms, and that's kind of the basis for modern taxonomy. For this reason, he's considered to be the father of modern taxonomy. So Linnaeus categorized organisms based on their morphology or their shape, and this is still being used today. The first 2 kingdoms that he came up with were Plantae and Animalia. But today we have all of these different kind of systematic taxonomy categories. Domain. Domains are, so there's 3 of them: Eukarya, Archaea, and bacteria. For the kingdoms, we have 5 kingdoms: Monera, Protista, fungi, Plantae, and Animalia. After this next exam that you guys take, we are going to spend the rest of the semester going kingdom by kingdom and investigating certain characteristics from within each one of these kingdoms. After kingdom, we have phylum, class, order, family, genus, species. So it gets more specific as we go down, less inclusive as we go down the list. So Carl Woese came up with it, 3-domain system: bacteria, archaea, and eukarya. And he did this based on sequencing of 16S rRNA for the bacteria and archaea and 18S rRNA for eukarya. This is a table of some distinctions between the 3 major domains of life. So as far as unicellularity go, bacteria and archaea are unicellular, while eukarya, some of them are unicellular, many of them are multicellular. You can see the difference in phospholipid structure, cell wall, nuclear envelope, membrane-bound organelles. You would not find them in the prokaryotes, but you would find them in eukaryotes. All of them have ribosomes. And then as far as introns go, some archaea have introns, all eukarya have introns. So archaea may be directly descended from and very similar to the first organisms on Earth. They're unicellular prokaryotes with distinctive cell membranes, as well as biochemical and genetic properties that differ from all other kinds of life. Some are autotrophic, which means that they make their own food, but many are heterotrophic, so they have to eat an external food source. They live in extreme environments. That's one of the characteristics that defines them. So they can live in hot springs or really salty lakes like the Dead Sea, in anaerobic environments, and so forth. The kingdom Eubacteria, in other words, just bacteria, are unicellular prokaryotes. Most of the bacteria that affect your life are going to be in Eubacteria. They're autotrophic and heterotrophic. There's disease-causing ones. There's ones that are beneficial. They reproduce by binary fission, and they have limited ways to recombine their genetic information. The kingdom Protista. So in our 5-kingdom system, both archaea bacteria and eubacteria are classified under the kingdom Monera. So when we get to Protista, we're continuing on with our standard 5-kingdom system. So these organisms are placed here more because of what they are not than what they are. So they don't really fit with all of the other ones, so we kind of just throw them here. Protista contains algae and protozoa. The algae are plant-like, and the protozoans are animal-like. They all have the typical cell structures that you'd find in eukaryotic cells, and these can be multicellular or unicellular. The kingdom fungi consists of the yeasts and the molds, and then there's like the slime molds and so forth. The cells of fungi have cell walls that contain the material called chitin. These organisms are heterotrophic, and they obtain their nutrients by releasing digestive enzymes into the environment and then absorb the nutrients from the environment. So that's considered being saprophytic. So they're going to release out digestive enzymes and absorb the nutrients. Plants are eukaryotic multicellular and carry out photosynthesis, and they are autotrophs. Many of them have cellulose in their cell walls. Plant cells are specialized for different functions, such as photosynthesis, transport of materials, and support. Plantae includes the mosses, ferns, cone-bearing plants or gymnosperms, and flowering plants or angiosperms. And then Animalia. So animals are multicellular, eukaryotic, and heterotrophic. They don't have cell walls. Excuse me. Most members of the animal kingdom can move from place to place, so they're motile. Some are permanently attached to surfaces, such as sponges and barnacles. Fish, birds, reptiles, amphibians, and mammals, including humans, belong in the kingdom Animalia. This kingdom also includes sponges, jellyfish, worms, sea stars, insects. We're going to go through all of the major phyla of Animalia and spend some time doing dissections on various species within Animalia. So looking at the classification hierarchy for numerous species, we have kingdom, phylum, class, order, family, genus, species, and you can see the names that exist here. Taxonomists split species into subsets called varieties. To classify organisms, modern taxonomists consider the phylogeny or the evolutionary history of the organism. Botanists sometimes split species into subsets, like peaches and nectarines are different variety of peach tree. Names were based on the Latin or ancient Greek words. The first word of scientific name is the genus, the second word is a species name, and usually genus is capital, species is lowercase, and then they're italicized. When we use the Latin name for an organism, like I said, we always capitalize the genus, and the second name, the species, is lowercase, and it's italicized. Systematics is a system that organizes the tremendous diversity of organisms into a phylogenetic tree. A phylogenetic tree is a family tree that shows evolutionary relationships that are thought to exist between organisms. And they have lines where different species will diverge from each other. So this is a phylogenetic tree, and we'll be looking at different phylogenetic trees and even creating one in the lab activity. The fossil record allows us to create this phylogenetic tree. And we talked about how fossils can be used in order to kind of support the theory of evolution. Morphology, or homologous or vestigial structures, that also allows us to classify and categorize organisms. And embryonic development. So again, we talked about that with the kind of evidence for evolution. If we look at the embryos, it allows us to create these phylogenetic trees. And then finally, chromosomes and macromolecules, we can sequence proteins and DNA and RNA in order to classify these organisms further. We can compare karyotypes. There's certain regions of banding patterns on the chromosomes that we'll look at. Human, chimpanzee, gorilla, and orangutan chromosomes are all lined up here, so you can see some of the similarities between the different chromosomes. Cladistics is a system of taxonomy that reconstructs phylogenies by inferring relationships based on similarities. It is used to determine the sequence in which different groups of organisms evolved. To do this, it focuses on a set of unique characteristics found in a particular group of organisms, and these unique characteristics are called derived traits or derived characters. So it shows, like this is a cladogram of the development of jaws, lungs, claws or nails. So it kind of shows when in an evolutionary timeline these different characteristics evolved. And again, phylogeny is the evolutionary history of a group. It's often represented as a phylogenetic tree, a diagram indicating lines of descent. Each branching point is a divergence from a common ancestor. There's ancestral traits and derived traits. So ancestral traits are present in all members of a group and they are present in the common ancestor, but they're not useful in determining the evolutionary relationships of ancestors or descents. Derived traits are present in some members of a group, but absent in the common ancestor, and they're the most important traits for clarifying evolutionary relationships. For example, an opposable thumb not present in the common ancestor of all mammals is an ancestral trait of primates. So here is showing a picture of phylogeny and common ancestors. So the fossil record is incomplete, like we keep saying. Many fossils fail to form or get destroyed. Homology is talking about looking at structures in different species with similar shapes or functions. Analogy is looking at structures in different species that do not describe an evolutionary relationship. So they kind of look alike, but they evolved by chance to look similar, but they don't have that evolutionary relationship. We can also look at behavioral traits. So in addition to molecular traits like we talked about with the DNA sequencing or protein sequencing, we can look at behavioral traits such as parental care, mating calls, etc. This is why animal behavior is so important.